Good morning, everyone, and thank you for joining us today. My name is Jibran, and I serve as the editorial member at the Journal of Immunotherapy and Precision Oncology, or JIPO. I'm excited to have the opportunity to interview Dr. Patricia Keegan. Dr. Keegan, would you like to kindly introduce yourself to the audience?
Thank you very much. Yes, my name is Patricia Keegan. I'm the Chief Medical Officer at Top Alliance Biosciences in Rockville, Maryland. We are the U.S. subsidiary of Shanghai Junshi Biosciences based in Shanghai. And I'm very happy to be able to talk to you today about our lead candidate drug, toripalimab, and our clinical study in patients with cholangiocarcinoma.
Thank you. We are happy to have you here. Dr. Keegan and her co-authors recently contributed an article to JIPO, which was titled Safety and Efficacy of Toripalimab in Patients with Cholangiocarcinoma, an open-label phase 1 study. So, Dr. Keegan, can you discuss the agent toripalimab and the rationale for its use in cholangiocarcinoma?
Yes, thank you. Toripalimab is an IgG4 kappa monoclonal antibody directed against PD-1. It was developed, it was developed by Shanghai Junshi Biosciences in China, and it has also been studied in the US in a dose escalation and cohort expansion trial, TAB 00101. And as part of that clinical trial, we evaluated the cholangiocarcinoma cohort, one of our larger accruing cohorts. What I would say about toripalimab is that it is directed against a unique epitope of the PD-1, distinct, for instance, from that is recognized by nivolumab or pembrolizumab. It has a slightly higher affinity for PD-L1 rather than Nivo or Ipi, and it has prolonged and sustained a binding to the receptor with internalization. So it has some unique features which make it a little bit different from some of the earlier PD-1 antibodies that have been approved. Our rationale in developing this, and particularly in the US clinical study, was to select cancers for which there was unmet medical needs, not adequate second or greater line therapy, in which we could study both safety and efficacy of this molecule in the US population. And we focused on cancer types which were known to be responsive to PD-1 inhibitors, which includes cholangiocarcinoma. At the time we initiated the study, for patients who had failed on chemotherapy, there was really no other approved or available treatment options. However, as we all know, during the course of the study, there was clear demonstration of efficacy of durvalumab in this population in combination with chemotherapy based on the TOPAZ trial. But at the time we initiated this, there was basically only some phase 2 data indicating that this was a cancer type which was likely to be responsive to immunotherapy. So that was our choice in selecting certain disease cohorts to further study our drug in this initial US trial.
Thank you for that introduction. Now, can you briefly review the safety findings and the anti-tumor activity of this drug in the cholangiocarcinoma cohort?
Sure. So in the cholangiocarcinoma cohort of 42 patients, we identified one partial and one complete responder among a fairly heavily pretreated population. So the population of patients who enrolled in this study were half of the patients had received between 2 and 4 prior therapies, and the remaining patients had received 5 or more prior therapies. They had received primarily chemotherapy. 100% of the population received prior chemotherapy, but they had also received a smattering of other treatments, including targeted therapies, and a few had received prior IO therapy. So this was a fairly heavily pretreated population. It had a slight female predominance. The majority were ECOG performance status 0. But among those, we did identify 2 responders, and we did show a reason with response durations of 4 and 7.8 months. And we did also show reasonable population with stable disease, approximately 40% of the patients experience stable disease. So we felt that this was evidence in this heavily pretreated population of some significant clinical activity, which would justify later trials. As I stated during the study, we did in fact see the results of the TOPAZ-1 study that also confirmed the activity of PD-1 inhibitors, PD-L1 inhibitors in this setting. With regards to the safety profile is very similar to other products in the class. The main toxicity with this product is immune-related adverse events. In this clinical trial, both in the cholangiocarcinoma population and in the overall population across all disease-specific cohorts, it was predominantly endocrinopathies being led by hypothyroidism, and laboratory abnormalities indicative of hepatic dysfunction. We did not see in this particular trial, in the cholangiocarcinoma, more significant evidence of colitis or pneumonitis or myocarditis. However, in our global development plan, we have seen those events with toripalimab. So it has a typical safety profile of an immuno-oncology agent. We saw fairly, because this was all monotherapy, good safety profile in that less than 5% of the patients experienced grade 3 or higher adverse events, and less than 5% required discontinuation of the drug for an adverse event. Approximately 15 to 20% did have dose interruptions for adverse events. And I'm sorry, I misspoke. Approximately 15 to 20% of the patients did have grade 3 adverse events. I misspoke there. But like I said, less than 5% required discontinuation of the drug, and 15 to 20% had temporary interruption for AEs. So we felt that this was a very good safety profile, particularly in the monotherapy setting.
Right, absolutely. Thank you for sharing the results. And so what are the future applications of this agent for this disease?
Right. So I think that the real, as with all effective agents, the real next step would be to look at the niche available in earlier line therapy. So we would like to bring this up either to the first line setting, or maybe even in this setting, it may not be possible to do an adjuvant or neoadjuvant or perioperative study as you would with the more common cancer, but to look at this in an earlier stage, and to look at it maybe head to head against some of the other anti-PD-1 agents. Sorry. In addition, we have received approval for toripalimab in the US in October 23 for the treatment of nasopharyngeal carcinoma in the first line setting, and have done a number of other phase 3 studies, primarily in China, evaluating this in combination with chemotherapy and a number of other solid tumors. So we do have a fairly good profile of that activity. But in addition to looking at this in earlier stage disease, first line, adjuvant, neoadjuvant settings, we're also looking to develop toripalimab in combination with other immuno-oncology agents or other effective agents. And so I think the natural next step is to look for development perhaps of chemo-free regimens, which might be more tolerable for patients, or combination with targeted agents such as an anti-VEGF agent, to see if we can bring this forward in a non-chemotherapy regimen, which again could be better tolerated.
Right, absolutely. Well, thank you very much, Dr. Keegan, for your time and your commitment to research.
Thank you.
Yeah, I would like to encourage everyone to explore the published article online at JIPO website. Once again, thank you.
Thank you very much for having me.